and thank you for tuning in. I am Dr. Al-Khalaf, a research fellow here at the University of Ottawa Heart Institute, and I am back as your host for today's episode of BEaTS Research Radio, a YouTube channel and podcast produced by the Bioengineering and Therapeutic Solutions Lab at the University of Ottawa Heart Institute. Our goal at BEaTS Research Radio is to communicate science to the community. Our guest today is Dr. May Griffith. Dr. Griffith is professor in the Department of Ophthalmology at the University of Montreal and researcher at the Maisonneuve-Rosemont Hospital Research Center in Quebec. Her research interests are in the area of regenerative medicine with special focus on biomaterials enhanced or enabled cell-based therapy. Dr. Griffith holds a Canada Research Chair Tier 1 in Biomaterials and Stem Cells in Ophthalmology, and she's the director of the cornea and anterior segment at the FRQS Vision Health Research Network. Dr. Griffith is a true leader in the field, advancing the use of biomaterials for the purposes of healing and improving patients' vision. Thank you so much for joining us at BEaTS Research Radio, Dr. Griffith.
Thank you very much for the invitation.
Absolutely, it's our pleasure. To start today, can you tell us how you come to focus your research on corneal abnormalities and diseases? What made you realize that there is an unmet need for research and development for solutions in this specific area?
Well, I started off actually working on nerves. So I love nerves, and the cornea is the most highly innervated organ in the body. So when I started, what I was trying to do was to do a much older version of what you call an organ on a chip for testing and toxicology. So one of the first things I did as a brand new naive professor was to phone the eye bank, and I asked the eye bank whether I could have, leftover donor corneas to use in my wound healing research. And the director of the eye bank pretty much laughed at me. Said, "What? Extra corneas? Do you not know that we have a shortage of corneas here and also worldwide?" And that's where I actually got into my research. So I had a wonderful director at the time, Dr. Bruce Jackson, and we sat down and we talked about, the different ideas. And of course, I'm a PhD. I'm not a surgeon, but my director at the University of Ottawa Eye Institute at the time is a surgeon. And so he ran me through the world shortage in corneas. He talked to me about places like LV Prasad and all the not-for-profit work that they're doing. And basically, I guess I got directed into this research, which I have continued ever since.
It's very interesting to know, and some of our audience might not know that cornea replacement is a very common and unmet need worldwide. And looking at the challenge from a global perspective, specifically curing corneal blindness in developing countries, what major hurdles or differences can you highlight to us to understand the variability between the corneal procedures in Canada and what's found elsewhere more commonly around the world regarding this health crisis?
Yeah, so in developed countries like Canada, most of your patients are the elderly patients. They would have, mild to sort of what we call medium risk for rejection. So be mostly keratoconus, some genetic conditions like Fuchs dystrophy, so which you can replace with endothelial lamellar keratoplasty these days. But in the developing countries, the factors causing corneal blindness are quite different. They range from infections, so really nasty infections, like we don't have trachoma here. They still have trachoma in lots of these countries. They also have severe injuries because there's a lot of industry and they don't have a lot of the safety standards we do. So chemical burns are quite common. We also have young children who are cooking. So when I went to India and I went to one of the eye banks there, they said that they do transplants on kids as young as 3 years old. And a lot of those injuries are from bursting spice packages or spices, because we don't think of food as being harmful, but there's capsaicin in some of these spices that are being used. So it's quite different. So there are 12.7 million people estimated globally to be on waiting lists for cornea transplants. And this does not include the other half of the world where there are no eye banks and no transplantation, and therefore not even a waiting list. Most of the severe conditions, so severe pathologies with inflammation, that affect more than the top layers, those are found in the developing world. And I have to tell you, one of the reasons why I got interested in looking at corneal blindness in the developing world was when I was a young child. I would travel with my parents. And one of the saddest things I found was a girl that was peering into the car I was in, and she had cloudy corneas. So I asked my dad at the time, why are her eyes so different? And my dad told me, well, there are many children who don't have enough food. And so, this was a vitamin A deficiency. And I draw, actually my sisters and I, we paint, we draw. And to me, it's very, very sad that children, or actually anybody else, would not be able to see all the lovely things that we have around us.
Yeah, of course. Vision is so important to connect to humanity and the world around us, and of course, anything that can benefit that would definitely bring some extra benefit to the world. And to that extent, the thought here is when the kind of work that you do in terms of biomaterials and maybe stem cell background research as well led you to the thought of using biomaterials as a replacement for human donated corneas. I wonder if you can talk to us a little bit about the differences between the artificial corneas that maybe can be synthetically made by biomaterials versus the human donor-based type of corneas.
Okay, so there are what we call artificial corneas that are now in clinical use. These would be the prosthesis. So their design and use is for replacement of very badly damaged corneas mostly. They are fully synthetic. So the best known one is the Boston keratoprosthesis. And that has a metacrylate optic in the middle, and it is sandwiched to a piece of human cornea. So the human cornea acts as a skirt, and this transparent optic is what transmits light into the eye to the back for vision. So that's the simplest form of artificial cornea. These are called keratoprosthesis. And the main aim there is to basically act sort of like a pinhole camera, and also as a protective shield for the more delicate inner portions of the eye. But it's not a replacement. So right now, the only widely accepted treatment are donor corneas. And as I told you, they are in short supply. So since I've always been interested in biomaterials, I thought, why don't we make some replacements? Why don't we just see if we can, regrow a cornea? So I looked at both stem cells and I looked at materials, and that's why I've been working in both these areas, because I think they're both very exciting areas and they have a lot of potential. So to make a long story short, I looked at what the components of the cornea is, and that's actually how you should be looking at designing things in the biomaterial fields. What is your target organ? What is it made of? How can you replace it? And then we thought, can we try to induce the patient's own stem cells to regrow instead of putting exogenous cells in there? Because it's the exogenous cells that the body recognizes as, outside. And then there's an allergenic reaction. So can we stop this? So we came up with trying to basically copy Mother Nature, because the human cornea is mostly collagen. So we thought the simplest way is to see if we can reproduce what Mother Nature has done. And so our first implants that we tested clinically were made from collagen. But we were also conscious of other things. So especially with COVID, you now know that there is a human animal transmission of pathogens. So back then it was not quite so recognized, but we were thinking a little further ahead. And also we had things like CJD, we had H1N1. We thought, no, this is not viable. We need something that does not fluctuate with, the year-to-year epidemic or whatever the disease of the year is. Or in the case of marine collagen, nuclear meltdown, as happened in Japan. So we settled at the time on recombinantly produced human collagen. We figured if it's made in the lab, it's going to be stable, it's going to be homogeneous, there's not going to be variation from supplier to supplier. And so we made our first implant out of recombinant human collagen. We did a whole lot of testing on animals. So we did mice, we did guinea pigs, we did rabbits. And then we had even 2 different subspecies of pigs. So we did a lot of testing. And of course, that before then, it was a pile of testing in the lab in vitro. And so we did our first clinical trial on 10 patients in Sweden back in 2008. So it's, more than 10 years now. And of course, every trial in an animal showed that we were able to get the tissues and to get the nerves to regrow. But of course, the big thing is it going to be the same in humans? And that's why we went through so many different species that I had some colleagues say, aren't you kind of overdoing it? But I guess, it's better to be safe than sorry.
Absolutely.
And, we couldn't sleep for the first little while when Per Fagerholm, the Swedish surgeon who actually did the trial, transplanted the first patients. So we were pretty much holding our breaths. And then, at 3 months, we saw that the patient's stem cells had grown in. They had proliferated. And basically, voila, we have in situ tissue regeneration in a patient's eye. And since then, we've repeated it in a second trial. At which point, the supplier for recombinant human collagen was not able to meet the supply, and then it was back to the drawing board. It happens. So now we're regrouping and hoping to redo our clinical trials again in Montreal and in Spain with the collagen analog.
Yeah, these are the kind of variables and issues that come up when it comes to research and development, right?
You don't plan for them, but it happens, right? So after 2 trials, you think, okay, we can now expand. And it's like, well, we don't have a supply of material, so it's back to the drawing board.
And I guess it's a good thing that this is in the biomaterials side of the equation, where there's some you definitely, you and your group and in general have the advantage of being able to pursue that. And actually, that brings me to the next question, which is, what do you see as challenges found on the road for clinical translation of biomaterial type research? Is there anything that can be done for maybe expediting the discovery and clinical translation of these types of uses for biomaterial sciences and stem cells?
I think one of the things that material scientists should be aware, if the goal is to actually go into the clinic. So while you can actually synthesize some really cool materials, the reality is that you have to get through the loophole, which is basically the regulatory approval process. And whether you like it or not, things that have already been approved for one use is easier to negotiate that hurdle than to design something absolutely new, like a new synthetic process. So for us, we chose recombinant human collagen because it had been tested as hemostats. And right now we are looking at collagen mimetic peptides. So we actually had our own designs, but the peptide we ultimately picked was designed by the Hetgering group at Rice University. And they had some data that it was good as hemostats, and they had, of course, their wonderful publication in Nature Chemistry. So it's a matter of what is your goal. So if it's goal to go to the clinic, then you look at whatever is best out there. And if it was to design something novel, which we also have, and many of our papers have not been translated, and I don't think they ever will. Like we had, synthetic fucoidants, we had some nice new glycopolymers, but basically it's to understand the regulatory process and to pick materials that already have been approved, maybe not in your area, but something else. So, for example, we're using a synthetic lipid. It has not been used in the cornea, but it has been used widely as stents, and it's been safe as coatings and stents. So it's a give and take. So
It's
you have to ask
definitely like
yourself, what is your goal?
yeah, and the need to like optimize from the very
Right.
beginning, optimize your road to translation by trying to see as much as possible what has been already tested and been proven to be safe. And I think these are some of the hurdles when it comes to approval for clinical use, is just knowing that there's a safety profile for something that is already in use, versus having to do all of the safety testing for something brand new that hasn't been at all, tested for those things.
Yeah, and then of course the other thing is to go on the websites of the regulatory agencies, whether it's the FDA, Health Canada, or the EMA, depending on where you're located, and start that dialogue early enough. So basically, before we've gone into our patients, we've started talking to the regulatory people. Like we've started talking to the regulatory people at Health Canada in 2015 for a trial that we had planned this year, which of course now is postponed because of COVID. But you start quite a number of years ahead of time. They're not going to tell you how to do your work, but they're going to, if you tell them, I'm going to do this, is it going to be acceptable? And sometimes it's yes, sometimes it's no. And in fact, back in 2015, it was, we would like to see this better tested in a more appropriate model. So what we did was we went back and developed a more appropriate model of inflammation. And we're hoping that next time we go see Health Canada, we can show them the results from the model that, when we had discussed, they said would be more acceptable to them. So you start early, you plan ahead. And the other thing, of course, is you work with a larger group of people. So doing everything by myself, I would be nowhere. Right. So I've always collaborated with a lot of people who are experts in their field. So basically, you put aside your ego. It's
The
like
value,
I have a wonderful
the
material,
value
but
of
somebody else would have a better idea. Yeah.
Absolutely. The value of collaboration in science nowadays is definitely it cannot be underestimated. And
Mhm.
this is the way to diversify and have an interdisciplinary group so that the work can be done again in an optimized and accelerated way.
Yeah.
Speaking of diversifying and using interdisciplinary and multidisciplinary approaches, stem cell biology and biomaterial sciences are normally seen as two separate fields of research. So I'm wondering, what overlapping themes were you able to identify on your way to developing your research path and career?
Well, actually, we know I worked on stem cells a long time ago when I was a graduate student. And back then, I was using different materials that come from the extracellular matrix, so like collagen, laminin, fibronectins, and I was using these to actually try and channel the stem cells to differentiate in different directions. So right from the start, it's always biomaterials for me have been a tool, and I have not been using plastic. So it's my materials are more biomimetic materials, so they're based on extracellular matrix. And if you're a cell biologist, these two have always been hand in hand, right? So it depends on what your training is. So for me, stem cells have always had a niche where they have been interacting with the materials in the extracellular matrix. So for me, it was not such a leap. It was just something that was very natural. I want something to regenerate. I should probably provide the stem cells the appropriate niche to come in and basically differentiate. So you
Yeah,
know
it's a
that's
matter of perspective, and it's good.
yeah,
It's really interesting that I mean I'm somebody that did a PhD in stem cell biology without
right.
ever really focusing on the fact that yeah, I do in fact have to coat the my plates with
Yeah,
extra and never really thinking of it as a biomaterials type of equation here, but it's a matter of seeing it from the perspective of the cooperation between these two separate areas to advance the goal here. So that's pretty. It's a good lesson to remember is that it's a science. You have to see it from the multiple perspectives that it is. And
yeah.
our last question today, Dr. Griffith, is what type of advice or words of wisdom would you like to share with early career scientists with respect to choosing their academic or career direction, either in the fields of regenerative medicine or wider field of biomaterial sciences?
I guess the main thing is to choose something that you like. So you have to like what you do because you're going to be doing this, at least for your PhD, your postdoc, or for the rest of your life. So it had better be fun, right? If it's not fun, then go do something else. It's uh, so that's the advice that I give to people in my group or people that I teach. You always have to like what you do. And actually, in some cases, I actually had a grad student whom I told him to maybe you should look at something else because this is not what I see you having fun with. You come in, you drag, and so he went into neural networks and he's just absolutely doing brilliantly. So he came back and he said, the best thing you did was to chuck me out of your group. But yeah, it's your career, it's your life. It's not what mom wants you to do, dad wants you to do, because I've heard a lot of that in my many years of being assistant director of grad studies at a program at University of Ottawa. So you follow your heart and be prepared to work really hard at it. So it is a career, it's not a job. So to go into research, it's not going to be 9 to 5. It's going to be, this is what I wake up to do every morning. If you find it's not what you want to do, then you should go into a different area. I mean, that's just common sense. Do what you like to do, and
Absolutely.
yeah,
Yeah, this is in fact, I call this like the golden piece of advice is realizing that, in science or in any career that you want to pursue, you have to remember that if there's no excitement, if you're not excited about what you're doing, it's always going to be a drag. It's always going to be more difficult if you cannot be excited by the results, by doing the experiment and seeing where the next thing to build on, comes from.
yeah.
That's exactly it. This concludes our interview. Once again, I'd like to thank our guest today, Dr. May Griffith, for the informative and fascinating talk with us regarding the therapeutic potential of biomaterials and stem cells in corneal development and diseases. BEaTS Research Radio is supported by the University of Ottawa Heart Institute, BEaTS Research Lab, and the Department of Biochemistry, Microbiology, and Immunology in the Faculty of Medicine at the University of Ottawa. On behalf of our director, Dr. Emilio Alarcon, and our regular crew of Haley Arnott, Alex Ross, and myself, thank you for tuning in. Don't forget to subscribe to our YouTube and podcast channels, and we'll see you soon again with another Science for the Community episode. Have a nice day.